Welcome back to Introduction to Biotechnology course. This is episode 6. Remember that you don't need prior experience, so let's start. What is animal biotechnology? It involves using animal genes for a specific purpose. Applications are mostly modern, unlike some older microbial techniques. Its goals fall into two main categories. Productive, creating something the animal doesn't naturally produce. Example, engineering a cow or chicken to produce insulin. Ameliorative, improving the animal itself. Example, modifying a cow's genes to increase milk production by 20 to 30%. You don't need to memorize the terms, just understand the ideas. Let's now explore the main applications. 1. Gene knockout, gene silencing. A molecular biology technique used to study a gene's function by disabling it. If we don't know what a gene does, we modify it to stop working. This modified gene is inserted into stem cells, which can form a complete embryo. These cells develop into an animal with all normal genes except the one we knocked out. If the resulting animal is born with no hair, for instance, we conclude the disabled gene was responsible for hair growth. 2. Cloning produces an animal without sexual reproduction. A somatic cell, any body cell, is taken from an animal. An egg is taken from another animal, and its nucleus is removed. The somatic nucleus is inserted into the empty egg, which is then stimulated to develop and implanted into a surrogate. The resulting animal is genetically identical to the somatic cell donor. Dolly the sheep was the first animal cloned from an adult cell. She had 3 mothers: genetic donor, somatic cell, egg donor, nucleus removed. Surrogate mother Dolly looked like the genetic donor. Scientists used different mothers to prove traits came only from the somatic DNA, not the egg or womb. Interestingly, Dolly died at age 6, half the normal sheep lifespan. Her DNA came from a 6-year-old sheep, so her biological age started older, shortened telomeres, linked to aging. 3-point CRISPR, a powerful gene editing tool, originally discovered in bacteria as a defense system. It's like using a highlighter and scissors on a sheet of genetic paper. You can find a sequence, cut it, and even paste in something else. Example, use CRISPR to remove the horn growth gene in cows, resulting in hornless animals. Difference between CRISPR and gene knockout. Knockout helps study gene function by turning genes off. CRISPR edits genes directly and precisely. 4. Transgenic animals. This means transferring a gene from one species to another. Example, insert a human gene into a cow so it produces human blood clotting proteins in its milk. Another famous case, inserting the spider silk gene into a goat so it produces spider silk protein in its milk, known as milk silk or biosteel. Spider silk is incredibly strong, stronger than steel by weight. This has uses in construction ropes, materials, and fine surgical threads. Why not farm spiders directly? Because they're cannibalistic and can't be raised in large groups, transgenic goats are a practical alternative. 5. Lab-grown meat. Also called cultured meat, it's made from animal cells in labs, no slaughter involved. The first lab-grown burger was created in 2013. It's valuable for ethical reasons, though still expensive. With more development, it may become commercially viable. 6. Xenotransplantation. This means transplanting organs between different species. Example, transplanting a genetically modified pig heart into a human, happened in 2022. Pig organs are similar in size and function to human ones, but their cells trigger strong immune reactions. Biotechnology helps modify pig genes to reduce rejection. The 2022 patient lived 2 months, a significant milestone. Though not a cure, it provided critical data for improving future procedures. These are the major applications of animal biotechnology. Each will be explained further in future episodes. Be sure to follow the sequence for deeper understanding.